So today, actually, we are going to be just talking about another sense, and in this case, we're going to be talking about vision. So far, we have talked about touch, and then during the next the next couple of days, then we're going to address vision, taste, and smell. That is between today and tomorrow. And I think the most remarkable thing is that when you look at this image, your visual system, your eyes, allows you to look at the different color, shades of blue. They allow you to look at the texture of those plants in that area. And it really allows you to look at the contrast in this image that I'm showing you today. And something that scientists have realized is that the eye is a very complex structure. Here I'm showing you eyes from different organisms. And I hope that what you can appreciate is that eyes, they come in many different colors, many different shapes, and all of them, they have evolved. Every organism has evolved an eye that allows it to see the best way that they can in their environment. And here I'm showing you different animals and how they adapt their visual system. There's this interplay between their visual system and their ecosystem. And evolution, as I said, just gets the best that they can. Here I'm showing you on the left side a bird, a predator, that is a diurnal animal, but has really high acuity and depth perception. And it will allow it to just eventually be able to grab the squirrel in the front.
What's the diurnal?
Do you guys know what diurnal means? If you don't understand something that I'm saying, please go ahead and ask me. A diurnal animal is an animal that the activity of the animal, basically the animal is awake during the day. And nocturnal here means that the animal is awake during the night. So if any of you have cats, you will notice that your cat is really lazy throughout the day. It's not really lazy, it's just that it's a nocturnal animal and they keep their activity throughout the night. Here I'm showing you a prey that it is a diurnal animal, and even the environment where they are, all these animals are land animals, but aquatic animals, they have to evolve a visual system that allows them to be able to see properly in an aquatic environment. You guys have any questions? And as I said, the eye comes in very different flavors. And there is something called binocular vision and monocular vision. When you hear these words, what do you guys think? What do you think monocular means and binocular means? Adara?
I think binocular is like two in the front of the face, and then monocular is on the side.
Yeah. So if you have to guess, what do you think that these animals are? This rabbit here, what do you think? Is it binocular or monocular?
Monocular.
What about the snake?
Monocular.
What?
Monocular.
You think monocular or binocular? I think binocular. What about this fish here, the zebrafish? Monocular. And this cat, cute little cat here?
Binocular.
And the horse?
Monocular.
And then the owl?
Binocular.
Okay. Yeah. So basically each of these animals, they have their eyes located in different regions. And Sadara said, some of them have binocular and other ones have monocular vision. And when I say monocular and binocular vision, what it means is that an animal has monocular vision. It has this region here, the field of view is increased. So now, imagine that you have your eye right here. You can see all this region here. And then if I turn around, now in the other side you will have, all that field of view. And there is a very narrow area that is binocular where both eyes are seeing the same image. And this, can definitely have several advantages, because if you can imagine what kind of animal between a predator versus a prey. Which one do you think is going to have monocular vision? The prey. Think about it. Which one do you think is going to have that monocular vision? The prey have monocular vision. Exactly. The animal has this huge area, a field of view that is just scanning to find that predator that is trying to eat it. On the other side, if you have binocular vision like this owl here, what it means is that the eyes are located more in the front, and they gave up their ability to have a wider field of view because now what they get is an increase in depth perception. So now the animal can determine exactly the location of their prey. And that's why I'm showing you here on owl. Do you guys have any questions about this? No? Are we good? Okay, because we're going to be doing actually some experiments on your depth perception later on at the end of the class. And I keep telling you about, the eye. So let me tell you a little bit more about the structure of the eye. So here I'm showing you a human eye. And you have a side view. There is a cornea, and that's that transparent part that is covering the neck structures that I'm going to be telling you about. You have an iris. This iris is what gives you the color. So some people have blue, green, and brown or dark brown. There's not such thing as a black eye. Let me just say that. And then what happens is that when the light comes in, it passes all these structures and the lens. What the lens do is that it focuses the light. That light then is going to hit the retina, and that's where the neurons are located. And I'm going to be telling you a little bit more about the structure in the next couple of slides. And eventually this light information then is converted into what kind of signal have we been talking about that the cells have been using over and over again. Those neurons that Evan and Victor told you about. Do you guys remember what signal the cell is using to talk to each other? Yeah, an action potential, but that action potential, it's made out of what? Electricity, correct. So then this information now has to be converted into something that cells can understand. And exactly that electricity involving an action potential that sends the information then to the brain for further processing. And this retina, this structure in the back of your eye, looks pretty much like I like to call it a cake. It has several layers that contains neurons. That would be what the cake is. And then the filling area is where those neurons, they convey with each other and they talk to each other in that region. So if you look at this eye, when the light comes in, hits the back of your eye, where the retina is located, and it has to travel all the way to the end, where these classical photoreceptors, rods and cones, are located. For these information to be meaningful, they have to detect it, and they have to change it from a light input into an electrical input. This light input then goes back here to these interneurons. And what the interneurons do is that they are modulating information. It's just they're making it sound bigger or smaller. That's what those neurons are doing. Then these interneurons pass the information to this group of neurons that are called ganglion cells. And the ganglion cells are the only cells that send projections to the brain. And then it will be further processed. Do you guys have any questions? So basically, the retina is the structure that detects light and converts that light into information that the brain can process. And if we look at those classical photoreceptors, the rods and cones, the reason why they're called rods and cones is because the cones look like a cone and the rods look like a rod. And they have very specific functions. The rods detect motion, and they detect dim light conditions. So right now your rods are not contributing to your vision system. Right now your rods are silent because we have a lot of light in this room. And that light's being detected by your cones, that they detect color and they also detect bright light conditions. Now, when we look at these particular photoreceptors, they have a very specific protein inside them. And that protein is called opsins. And there are many different kinds of opsins. There's rods that they detect, as I said, the dim light conditions, so there's not a particular color associated with it. But then you have red, blue, and green opsins. And what all these opsins do is that they're going to detect light at a very specific wavelength. So basically, light is composed of many different colors, and when that white light is breaking down, then you can start seeing the blue, the green, the red, and that's what these specific proteins are doing. And their location of these cells in your eye is very important. Here I'm showing you the back of that eye. You have a lot of different veins that are just providing nutrients and oxygen to those neurons. And you have, when you look at it, you have all of these are rods cells, and they're all everywhere except in the fovea. And then in the fovea, you have many, many, many cone cells. It's just devoted to cone cells. So what that means is that then you have different kinds of vision. There is peripheral vision, and that means that these rod cells are detecting light, and that's why rods detect motion, because you're detecting it with your periphery. Now, if those cells were damaged, what happens is that this is what you will see in a normal image. But when the rods are damaged, then you can only see what it is in the fovea, in those cones, and that's what the image will look like. Now, what do you think it would happen if the central vision is damaged? What do you think that those cone cells will be detecting? How this image would look like? Do you have any ideas? So if the periphery is damaged, you lose the periphery here, right? So now if the central side is damaged, what would you be looking at that image?
It would be blurry in the center, or you wouldn't see the center.
You wouldn't be seeing the central, it will be blurry. Exactly. So now if you look at that, that's exactly how the Mona Lisa would look like. The center part of Mona Lisa would be missing, but you will be able to see the rest because your periphery is the one providing that information. You guys have any questions? Okay. And as Evan and Victor has been telling you about, there are animals that have amazing vision system. So do you guys recognize all the animals in this image? So what up? This is the easiest. What is this? A what? No. Think north. No. Think northern than that and think about December.
Reindeer.
A reindeer, exactly. What about this one here? This is a tough one. No, it's actually a goat. And this one here? It's a dragonfly. And what about this? It's a fish, right? So actually this is a goat, this is a dragonfly, and this is a four-eye fish. And what these animals have in their vision system is that the reindeer can detect UV light. That's something that we cannot do, of course. The goat, do you see here the pupil? It looks like a square. So the goat can constrict, instead of constricting the pupil to look like a circle, they constrict it as a square so they can control way better than us the amount of light that goes into the eye. And
Is it similar to cats?
no, cats will still get the circular, so goats are way better at controlling the amount of light. What about this? Do you guys know what kind of eye is this? When you look at that eye, what do you see? What do you think that you see? What? Different colors you see? So if you look close enough, what you see is that they are tiny little lenses. They're tiny little, let's call it mirrors. That's how I see it. And what it is, it's a compound eye. Do you guys have you ever heard about a compound eye? No? So the dragonfly and most insects, they have tiny little eyes that each eye sees a little tiny piece of the image, and then they combine all that information in the fly brain and allows it then to form an image. So this particular one has between 5,000 to 6,000 small eyes. And what do you think about this? What do you think about this name, four-eyed fish? You think do you see four eyes in there?
I don't.
You don't? Yeah. So what this happens is that they call it the four-eyed fish because the eye has right in the middle has a tissue that divides the eye, and then you have the top of the eye, it's on the top of the water, and the other side of the eye, it's underneath the water. Now the animal can look at what is happening above the surface and underneath the surface. So it can make sure that nothing will eat that fish. And that's why I think it's a pretty cool animal. And they definitely have very different light. So there are different ways of how we can detect or test for vision. All of you have gone to the doctor and have this particular vision test. And we cannot ask in the lab, hey, mouse, can you read this book for me and let me see whether or not you can see? What we have to do is that we place animals in this arena, and we rotate these bars, and then we look for the animal to move the head. As the animal moves the head, then we can determine whether or not the animal can see it. So if the animal doesn't move the head, the animal can't see. And we can even test for your ability to see color. So can you guys tell me what this is, the number on top?
29.
The one next to it?
29.
And the next one?
45.
And the next one?
56.
And then the next one?
6.
And the next one?
8.
So if you couldn't see those numbers, you probably already know that you're colorblind. And this is actually what a colorblind person would be able to see. If we look at the statistics of colorblind, 10% of the males in this room are colorblind. They're not 10 males, so I'm not sure if we have a colorblind person here. But it is extremely common, and it's really rare to find a female that is colorblind because you need two genes inherited one from your mother and one from your father of the colorblind. Now, there is something called optical illusion. And I'm going to be telling you about it. But before that, I want you to look at that flag. And I want you to focus your eyes. I don't want you to be able to do the microsaccades, meaning move the eyes a little bit. And after 15 seconds, between 15 to 30 seconds, I want you to then look at that cross there and tell me, what do you see? So I'm going to start counting. 1, 2. Look at the cross. What do you see? Do you see something?
Yeah, you see like the American flag and like the right colors.
You see the inverted flag with the right colors. Did everyone see that? Now? Okay, we're going to have more exercises.
Let's try it again.
Yeah.
Okay. You cannot move your eyes. So just focus and try to keep as steady as you can. Do not move the eye. It's going to be really hard for you to keep your eyes locked in one position, but just do it.
Gabriele,
Daniela, do you want to sit on this side?
Yeah, you probably want to sit in the middle. It will help.
Now look at the right.
Yeah, I can see that.
Can you see it better? Val, can you see it? Yep. Okay. So what that is, it's called the after image effect. And what just happens is that your eyes just got overstimulated. And when that happens, your photoreceptor cells, they get adapted. And once they get adapted, if you move your eye, you're going to be able to see. Basically, the cells that got adapted, they got tired. Because they got tired, you're not going to be able to see those colors, and you're going to see the complementary colors, meaning the other photoreceptor cells will be kicking in. And that's what the after image effect is. And it happens all the time. If you look at the flash of a camera and then look to the side, you see this blurry thing like floating around, your eye just got they got overstimulated and they got adapted. What about this one here? Look at the bird. Now look at the cage. What do you see? Now? You want to look back again? Look at the bird. Now go back to the cage. Okay. And it's the same concept. It's an after image. Now, this is the lilac chaser. What I want you to do is to look at the cross right in the middle and then tell me, what do you see? Does everyone see the green dot? Okay. So the lilac chaser was actually created around 2005, and it was actually a mistake. The person wanted to design a different experiment and ended with this one, and it worked well, so stick to it. And basically what is happening is that there is an empty space in between. There's an area that there's no magenta circle, and your brain just fills it up with the complementary color of magenta, which in this case is green. Now, tell me, is that orange circle on the left and the right, are they the same size? Is the one on the right bigger, or is it smaller? You think the one on the right, it's bigger? How many of you guys think that the one on the right is bigger? How about the same? Smaller? Okay, good. So it gives you the illusion that the one on the right side is bigger, but it's actually equal. And this is what it's called a relative size perception. What it means is that when we are comparing sizes, when we are doing a comparison, we always compare it to something else. The circles on the left are definitely bigger than the one in the middle, and that's why you think that it's smaller when you compare it to the other side. Now, I want you to look at this image and tell me immediately, what do you see? Just shout it out, okay?
Two faces.
Okay, how many of you saw the chandelier or the cup, however you want to call it? Okay, how many of you saw the face? Okay, and this is what we call, what you're looking at, it's just perception. Your brain looks at those lines, at the contrast of the image, and then based on previous experiences, decide what it is. And it compares backgrounds, the surrounding, and that's why sometimes you can see the vase, and other times you can see the faces. There is studies in psychology where they present these images, and then they ask the person, what do you see? And based on your answers, they may conclusions of your personality. And I will not even start to address that because I really don't understand it. Yeah, and if you go online, you will find many of these images that some people can see one thing versus the other. So now what we're going to do is that we're going to split, just find a partner and go into one of the tables. And what we're going to be looking at is at depth perception.